Alrighty, y'all. Today we are going to be learning about the Krebs cycle. So if you remember, when we left off with glycolysis, we produced some pyruvic acid, otherwise known as pyruvate. And that is going to be one of the first things that we start with in the Krebs cycle, right? It gets sent over here as something that we can then immediately utilize. But before we jump into the Krebs cycle, we have to know more about the actual mitochondria. So, first thing that we want to know, the mitochondria is a little funky, right? There is something called endosymbiont theory, which tells us that perhaps the mitochondria may have once been its own individual prokaryotic organism that was swallowed up by another eukaryotic organism and turned into an organelle, which is really, really weird. And we have some evidence for this, right? We don't just think that because it looks cool. We actually have some stuff that tells us this. First thing is that the mitochondria has circular DNA. Right? It has circular DNA, its own set of DNA, different from the linear chromosomes that we'll find in the rest of the eukaryotic cell that it is inside of. Right? It has in that DNA genes that can code for all sorts of different things. Right? There are 37 genes in the mitochondria. These genes are going to code for the following things. Right? We can code for 13 different proteins. Remember, our DNA codes for proteins that we can use in order to build up the rest of the parts of our organism, right? So all of the parts of our cells, all of the different organelles, every molecule is built from all of this. What we're also going to see is that there are 22 genes that help us code for different tRNAs, right? The transfer RNAs. These are the things that carry our amino acids to assemble them into proteins, right? So it already has the stuff it needs to start assembling the proteins that it's making, and it has 2 rRNAs being coded for, right? These make up our ribosomes, the actual factories that'll put those amino acids together. So we can produce the amino acids and then assemble them into the proteins at the different places that we build in our ribosomes, right? This has everything it needs to build its own structure, right? So this is going to be evidence for endosymbiont theory. Endosymbiont theory. That is ENDOSYMBIONT. Endosymbiont theory, right? And endo means inside, like an endoskeleton is what you and I have, whereas an ant would have an exoskeleton, and then symbiont, like symbiosis. So this mitochondria was pulled inside of another eukaryotic organism and then utilized in a symbiotic relationship where it would produce energy and it would not get eaten and digested by something else, right? Instead, it is protected inside of this other organism. So the mitochondria is important for a lot of different reasons. I'm not just talking about it because it's cool, even though it really is, right? The powerhouse of our cell, and it's maybe its own weird extra organism inside of everything, but it has a lot of different pieces to it, right? We have ribosomes in our mitochondria. And again, remember, these are for protein synthesis. So building our proteins, everything that we build to make our structures, not only do we have the genes to make it happen here and the stuff to start building everything that happens here, but we have the factories, the protein factories, our ribosomes are here as well. We also have a lot of different pieces of the mitochondria out and about around it. So we have this outer membrane surrounding it. The outer membrane surrounds the entire mitochondria. But then note, we have this twisted up, weird, curly Q thing going through here. This is another membrane that's folded up and shoved inside of that outer membrane, right? This would be the inner membrane. And this combination of an outer membrane and an inner membrane is another piece of evidence for endosymbiont theory, right? It tells us Well, this organism had the inner membrane, and then it was swallowed up by something, which then encapsulated it in its own membrane when it did, right? Remember vesicles, when we have our endocytosis and we're bringing something into the cell, we surround it in a membrane. That's what we think happened to give the mitochondria its own second outer membrane. And then there's the space between these two membranes, very creatively called the intermembrane space. And lastly, what we're going to talk about today is something called the mitochondrial matrix. And I'm not talking about like the movie with Neo and going to find out that he lives in, spoiler alert, a simulation, right? Instead, I am going to be talking about the mitochondrial matrix, which is this fluid, gel-filled space inside of the inner membrane. So that's this arrow right here. I don't know why there's two arrows pointing off of it. This one's pointing to the outer membrane again for some reason. We're going to cross that one out, right? We're looking at this space inside of our inner membrane where all of this, the ribosomes are floating around and our circular DNA is found, right? This intersection. So this right here, this mitochondrial membrane, again, it's this gel-filled space inside the inner membrane. And not just a cool movie. And this mitochondrial matrix, right? This space in the inside the inner membrane is where the Krebs cycle happens. And that's what we're going to be talking about today. Right? This is going to happen right here in the mitochondrial matrix. So, let's look back here. We're going to kind of refresh. We've talked already about glycolysis. We said it produces 2 net ATP, right? We use 2 ATP and then we produce 4. So minus 2 plus 4. We now have positive 2 ATP produced. Great, we're making some energy. The Krebs cycle is going to take some of the stuff from there and produce 2 more ATP. And then tomorrow, we'll talk about the electron transport chain, and this is where the majority of the ATP that we produce comes from, right? Here, we get 32 more ATP. So all of these, right, are part of cellular respiration. In total, cellular respiration produces 36 ATP. But we're just talking about this part, the Krebs cycle, which produces a lot of the stuff needed to get to the electron transport chain, right? The electron transport chain, that's our main deal. That's where the vast majority of our ATP comes from, where all this energy is. But to get there, we need electron carriers for the electron transport chain, which we are going to make in the Krebs cycle. That's the whole goal of the Krebs cycle, is making electron carriers, right? So we start with pyruvic acid from glycolysis. It comes down into the Krebs cycle, and here in the Krebs cycle, we'll produce our 2 ATP, get a little bit of energy out of it. That's pretty nice. But the main thing that we're here for is this NADH and this FADH2. These things are both electron carriers. They are just like our NADH plus, right? Or the NAD plus, which, my goodness, the NADP plus, which becomes NADH. I cannot speak for the life of me. The NADP plus, which becomes the NADPH, is very similar to NAD plus, which will become NADH, right? They do the same job. They are electron carriers. They're going to carry electrons. It's just they carry them to different places. NADPH will carry electrons in photosynthesis. NADH is going to carry electrons here in cellular respiration, right? The counterpart of photosynthesis, which does much the opposite. It produces energy using glucose and using our oxygen, and it produces carbon dioxide, and it produces water, and it produces more high-energy molecules, ATP. So it's much the opposite. Ooh, that was a tongue twister. NADH and NADPH. Very similar molecules, very similar names, too. They both stand for nicotinamide adenine dinucleotide, and then pH has phosphate in it. NADH is just nicotinamide adenine dinucleotide no phosphate in there, right? They just both have that extra hydrogen group on them. So now, and again, you don't need to know those names. I just like the long names. So now, we are going to talk about the Krebs cycle here, right? So at the end of glycolysis, like I was saying, we're going to end up with 2-pyruvic acid, which are these 3 carbon molecules, right? We took a glucose, we split it in half, right? We still have a lot of energy available to us. We need to extract that from these pyruvic acids, aka pyruvate, right? So these pyruvic acids are going to go to our Krebs cycle. And that Krebs cycle, once again, happens in a place called the mitochondrial matrix. But we only go to the Krebs cycle as long as we have a bunch of oxygen. If we don't have our O2 here, right, then we're going to undergo fermentation after glycolysis and just keep producing a little bit more ATP there. But our end goal is the electron transport chain, so we'd much rather have oxygen and then be able to produce this huge amount of energy from our electron transport chain here. So we need to go through the Krebs cycle to produce these electron carriers. What we're going to see, right, pyruvic acid, pyruvate still has about 90% of its available chemical energy, right? As we go through cellular respiration, most of the energy, roughly 62% of the energy, is going to be lost in the form of heat, right? I'm going to write that down. Around 62% of our energy in cellular respiration is lost as heat. Just a really, really cool thing to know, right? Most of the energy is going to be wasted, sent out into the atmosphere. And the reason for that is because we are taking these high energy molecules, right, these electron carriers, and we're breaking the bonds to release that energy. And when we break the bonds, when we release the energy, a lot of it is just released into the environment, right? It gets warm, it gets released out into the environment. That's why when we exercise, we heat up, is because we're breaking all of these molecules as we go through these processes, and they are releasing energy and heating us up. So what we're going to see, though, is that a lot of the chemical energy at this point is still unused, right? 90%. We haven't wasted all of this as heat yet. We're going to waste some as heat along the way, and most of the energy, though, that we don't waste is going to end up in these electron carriers so we can send them over to the electron transport chain. So that's what we're going to look at here now on the backside, is the actual Krebs cycle. So we start off here in step 1. I'm going to label this box as going to be all of our step 1 stuff happening here. So in step 1, we start with pyruvic acid, right? There's 2 of them. We split a glucose into 2 pyruvic acids. Note, this diagram is only showing the Krebs cycle as we go through 1 pyruvic acid, right? We are going to actually repeat this whole cycle twice for every single glucose that we are spending through glycolysis, right? So we start with 1 glucose, and then that goes through glycolysis, produces 2 pyruvic acids, so we can then do the Krebs cycle here twice. So what we're going to see, that pyruvic acid enters the mitochondrial matrix where our Krebs cycle can happen. One of the carbons is going to break off of that pyruvic acid, right? It's a 3-carbon molecule. We're pulling 1 off, boom, to make carbon dioxide. That's released out, right? But, so that's one of our products. We've already produced one of our products, carbon dioxide. I'm going to be highlighting stuff as I go. So carbon dioxide is going to be in blue here. We have made one of our products. So I also, in the process of breaking this bond, release some very high energy electrons, right, out of my pyruvic acid. So those high energy electrons are then going to be snagged by an NADH, right? This, or it was an NADP, or an NAD plus, it becomes an NADH, which is an electron carrier. That's another one of our products here, what we're actually trying to produce with the Krebs cycle, right? We need to carry these electrons over to the electron transport chain. Thus, we need electron carriers to take them over to the electron transport chain. I'm going to do ETC equals electron transport chain, right? So they are taken over to the electron transport chain here. And again, that's my NADH, this high energy electron in my electron carrier. I'm going to label that here. NADH is an electron carrier. This is our goal for the whole Krebs cycle, so it's really important that we know this. NADH is an electron carrier. It is, and FADH2 is also an electron carrier. So I'm going to highlight that in yellow. So now we have one of our electron carriers already produced. Fantastic. We're pretty happy with that. Now, we pulled that electron away by breaking the carbon dioxide off of a 3-carbon molecule, right? We took 1 carbon away. Now, we're left with 2 carbons, right? I have 2 carbons. That's these 2 right here, formed up in a 2-carbon molecule called acetyl CoA, or CoA, right? You don't need to know that now. It's a 2-carbon molecule. But CoA is also the name of the enzyme that we're going to need for our next step, right? CoA, right here, coenzyme A is what this stands for, right? This is the enzyme needed for what's about to happen next, right? So we are going to take these 2 carbons from this molecule, and we are going to combine them together with a 4-carbon molecule. Right? So now we have 2 carbons plus another 4-carbon molecule, which is this one right here from earlier on in the cycle, right? We're going to reproduce this 4-carbon molecule in our next step here so we can keep the cycle going forever, right? That's how cycles work. We want to keep them going. So I'm going to take a 4-carbon molecule, I'm going to take a 2-carbon molecule, and I'm going to make citric acid out of this. Citric acid is a 6-carbon molecule, right? So 2 plus 4, we get 6. Some crazy math, I know, but we got to learn something hard today, right? So this enzyme, CoA, right, coenzyme A, is the enzyme needed to make our citric acid. So here, I'm going to highlight my citric acid in green, just to keep my colors varied here. So here's my citric acid, right? Citric acid, which is a 6-carbon molecule. So this is actually where the Krebs cycle gets its other name, right? Krebs was the name of the guy who won the Nobel Prize for discovering this whole thing, right? Hans Krebs back in 1953. But what we're going to see is that sometimes we don't want to just talk about it when we're referring to the name of some old white guy who it was named after a long time ago. Instead, we also want to talk about what is made and get technical with our names because science loves technicality and nomenclature. So we also call it the citric acid cycle because we make citric acid as part of it. And then we're going to break that citric acid down. It's not really one of our products. The citric acid cycle is just saying we're going to have citric acid as part of this, right? We're actually going to have 4 end products here, which I will get to at the end as we go. But we already know 2 of them. We've already produced NADH and carbon dioxide. Those are 2 of them. We'll label the other 2 as we find them here in our cycle. So that's step 1, right? And just to reiterate, we start with pyruvic acid, this 3-carbon molecule. We rip a carbon dioxide off of there. That releases a high-energy electron, which is dragged away as NADH to the electron transport chain. That leaves us, right, 3 carbons minus 1. We have a 2-carbon molecule, which this CoA, coenzyme A, is going to help connect to a 4-carbon compound, and that's going to make a 6-carbon molecule called citric acid. Start with 3 minus 1. We have 2. Add 4 to it. We get 6 carbons. Lots of different numbers of carbons and things. This is why we didn't do this at the same time as our photosynthesis unit, because otherwise we would all be confusing this and the Calvin cycle together, and there is no reason for that level of confusion, right? Let's do them one at a time. Now, next step, step 2 here, right? So that's everything in this rest of the circle. So step 2, we have citric acid, and we're going to break it down. And I'm not saying we're going to like break it down like I'm going to break into song here now, though I absolutely could and am tempted to, but I'll spare you all that today. So we have citric acid. We're going to further break it down. When we do so, the first thing we're doing is pulling one more carbon off of it, getting more carbon dioxide. And just like last time, by pulling that carbon dioxide off, we end up releasing high-energy electrons, which are going to form into some electron carriers. But this time we're going to get 2 electron carriers out of this, right? We're going to do this a couple times, so we're going to get NADH, but we're also going to get FADH2. Right? So we're going to have these electron carriers now. So just to reiterate, we release carbon dioxide. Here I have it right here. When I did that, I'm going to get high energy electrons to form NADH and FADH2. And again, all these things in yellow, NADH and FADH2, these are electron carriers. These are what we're trying to produce here so that they can carry these electrons over to the electron transport chain. But this is the goal. We're accomplishing it. So here I can see I have produced NADH, right? And I now have a 5-carbon compound. And then I'm going to rip another carbon dioxide off of it, right? This step 2 is kind of like just happens over and over. I rip carbon dioxide off of that. And this time, when I do, I'm going to end up producing NADH. I'm also going to get some ATP out of this. I'll do that in orange here, right? I get some ATP, more high-energy molecules that I can now go spend. We love our ATP. It's money, right? And then, by doing that, right, my 5-carbon molecule, I pulled a carbon off. Now I have a 4-carbon compound, which, if you remember, that 4-carbon molecule is then going to be combined with the 2-carbon molecules from up here in order to make our citric acid, so the cycle can repeat, right? But to get it there, we're going to be able to pull some stuff off of it first before it combines. We can pull some high-energy electrons out of there, and we'll end up with some more FADH2, some more NADH. Look at all these electron carriers we're getting out of here, right? We are producing so many electron carriers. That's the main thing that we produce here. So now I know my products here. I have made some ATP. Remember, we're going to do 1 ATP from this cycle, but I'm going to repeat the cycle twice for every single time I do glycolysis. So I'm going to get 2 ATP as my product up here. And then the other electron carrier that I've produced here is going to be FADH2. So I have now produced my 4 products of the Krebs cycle, right? NADH, CO2, 2 ATPs, and some FADH2. So now all of the energy that we've gotten from our Krebs cycle, right? All of it has gone into the form of some high energy molecules, right? We have ATP that we produced, but we have also produced these NADH and this FADH2, these electron carriers. So the ATP we can use immediately, right? That's what the cell needs to go do its job. Remember, it doesn't last very long. We got to go spend it. It's the currency of the cell. It's not something you're going to save in your bank account. It's no gold bar, right? It's losing value fast. So you got to go spend it. I'm not telling you to go spend all of your money, though. Save money. Make good financial decisions. So what we're going to see instead now is that these electron carriers NADH and FADH2, right? These are going to go over to the electron transport chain. But these are the main goal, right? The electron carriers, I'm going to put some stars around it. I'm going to highlight this in yellow like I've been highlighting all of my electron carriers so far, right? These are the main goal. I'm going to put that over here. I'm even going to write that. These are the main goal of the Krebs cycle. So we don't do this to get the ATP. The ATP, very nice, side benefit, right? This is not why we did the Krebs cycle. The reason we actually did the whole Krebs cycle is for these, right? NADH, FADH2, electron carriers. I'm going to highlight them in yellow every time I see them. They're important. NADH, FADH2, electron carriers. And where do they carry these electrons? That's right, to the electron transport chain. Did you like the Dora the Explorer moment there? So, right, what we're going to see now is ATP. We get a little bit of it here. Fantastic. We can go spend that. But now we're going to take these electron carriers over to the electron transport chain, and that's where we are going to produce the majority of our ATP, right? In glycolysis, we netted 2 ATP. Here, we netted 2 more ATP. That is 4 ATP total out of the 36 that we are going to make. So the next 32 ATP, the vast majority of it, we need the electron transport chain to make. So that's what we're going to talk about tomorrow. Today, what I want you to do with the rest of your time is to work on this assignment here that I'm going to have passed out to you. This assignment, you are going to get through problems 1 through 7, and then I believe it was problem 15 as well that you can answer, but you won't be able to answer about half of the problems on the assignment, right? Because those are about the electron transport chain, which we won't have learned about yet today. So use the time that you have wisely. We should still have about half an hour in class at this point, so I want you to use that time. There's really only 8 problems for you to solve, and once you finish, hold on to the papers, because tomorrow we'll learn about the electron transport chain, and then we'll finish this worksheet. But today, I want you to just do these assignments, and then with any remaining time you have, take some time, study over this diagram as well as what we learned from glycolysis, because we will be having a test on this material quite soon. Again, this is one of our really, really small units, and I need you to be prepared for it. And you remember just how difficult these diagrams can be, so make sure you're putting the proper time in to actually learn them. And if you feel confident about all of that, do it again. And then and only then, I want you working on something else from your other classes, or just don't make it a social hour, right? Be kind to my substitutes. They are working hard here, and they don't know you like I know you, and they don't know that your intentions are good when you act the way that you act, so be kind to them. Okay, thank you, everybody. I'll see you tomorrow when I am back from my meeting here. Now, if I can just get the darn mouse here to work. Why did this die now, of all times? Oh, there we go. There we go. Okay. I'll see you tomorrow.